Our Earth is home to countless plant and animal species, all of which have a limited lifespan and are bound to die. Yet these species have existed for several thousands of years. That's because living organisms can reproduce. That is, they give rise to offspring, which in turn grow and reproduce their own offspring of the same kind. This ensures survival of the species generation after generation. In living organisms, we see two types of reproduction: sexual and asexual. In sexual reproduction, a gamete from the male parent fuses with a gamete from the female parent to form a zygote. However, in asexual reproduction, the organism or the parent cell divides to give rise to morphologically and genetically identical organisms called clones. Asexual reproduction, commonly seen in protists, moneruns, and fungi, is of many different types. The amoeba, for instance, reproduces by binary fission, where the parent cell divides into two halves and each half develops into a daughter cell. Likewise, bacteria too reproduce through binary fission. Yeast, on the other hand, reproduces through budding, where the parent cell produces small buds through mitosis. These buds initially remain attached to the parent cell and then separate to develop into new organisms. While yeast reproduces through budding, other members of the fungi kingdom, such as penicillium, reproduce through conida, a special asexual reproductive structure. Apart from fungi, we also see asexual reproductive structures in certain members of kingdom Animalia. In the case of Hydra, buds which develop on the parent body serve as a means of reproduction. Likewise, the sponge produces internal buds called gamules that aid in asexual reproduction. Apart from single-celled organisms and animals, asexual reproduction is also seen in simple plants such as algae, as well as in certain higher plants such as potato. For instance, Chlamydomonas, a type of algae, develops zoospores which later mature into new plants. However, during unfavorable conditions, Chlamydomonas, as well as other types of fungi and algae, undergo sexual reproduction. In higher plants, asexual reproduction, commonly known as vegetative propagation, takes place through vegetative propagules, specialized structures which emerge from different parts of the plant. For instance, the stem of a potato plant is dotted with buds known as eyes, which later germinate into new plants. Whereas in a ginger plant, modified stems or rhizomes having nodes and buds act as a means of vegetative propagation. While the buds develop into a new ginger plant, the nodes of the rhizome give way to adventitious roots which help in the absorption of water and minerals from the soil. Bryophyllum, on the other hand, have notches on the margin of their leaves that give rise to adventitious buds that fall off and later germinate into new plantlets. Apart from buds and nodes, runners, suckers, offsets, and bulbs are other examples of vegetative propagules in plants. Interestingly, farmers and gardeners make full use of vegetative propagation for commercial cultivation of various plants, including strawberry, potatoes, and ginger. Vegetative propagation, however, can turn into a problem in certain cases, like the fishermen of Bengal have discovered. water hyacinth, an aquatic plant with a phenomenal rate of vegetative propagation, is choking the Bay of Bengal and killing millions of fish, thereby ruining the livelihood of the fishing community. Asexual reproduction, an important biological process that ensures continuity of life, occurs differently in different organisms. Reproduction, an important biological process occurring in living organisms, ensures continuity of life. Organisms reproduce either asexually or sexually. In the case of sexual reproduction, we see the fusion of male and female gametes, which are either produced by the same individual or by different individuals of the opposite sex. Though organisms which engage in sexual reproduction vary in their external and internal structure, the pattern of sexual reproduction remains quite similar. All organisms, for instance, go through 3 phases of life: the juvenile phase, reproductive phase, and senescence phase. The juvenile phase is a period of growth which is characterized by an increase in height in all animals, including human beings. Whereas in plants, we see the emergence of new leaves during the juvenile phase. Did you know that in plants, the juvenile phase is known as the vegetative phase? Interestingly, the duration of the juvenile or vegetative phase varies in different organisms. In human beings, for instance, the juvenile phase lasts for 13 to 16 years in the case of females and 13 to 15 years in males. Whereas a mango tree remains in the vegetative phase for around 3 to 5 years. The juvenile phase is succeeded by the reproductive phase in both plants and animals. In the case of plants, this phase is marked by the appearance of flowers or vegetative propagules such as runners. However, while some plants such as the rose flower throughout their lifespan, others such as the mango flower seasonally. Interestingly, in the case of mango, which also happens to be a perennial plant, it is very difficult to define the various phases of life. That's because perennials, which have a long reproductive phase, keep shedding their leaves throughout the year or once a year in winter. The shedding of leaves can be misinterpreted as a sign of the senescent phase. However, it is easy to identify the phases in the case of annuals and biennials. As all 3 phases of an annual plant occur in 1 year, whereas biennials have a 2-year life cycle in which the plants enter the vegetative phase in the first year and the reproductive followed by the senescent phase in the second year. Did you know that certain plants such as bamboo flower only once in 100 years, produce fruits and then die? Similarly, Strobilanthes kuntiana or Neelakuranji flowers every 12 years. Just as plants develop specialized structures during the reproductive phase, animals too show certain morphological and physiological changes. In human beings, for example, the reproductive phase, which starts with puberty, leads to the development of male and female secondary sexual characters such as facial hair in male and breasts, which are seen more prominently in females. Moreover, females of all placental mammals, including primates and non-primates, demonstrate cyclical changes in the activities of their ovaries and accessory ducts, as well as hormones during the reproductive phase. Known as the menstrual cycle in primates and the estrous cycle in non-primates, these cycles stop once the female becomes pregnant and resume after the female has given birth to offspring. Moreover, placental mammals, especially those living in the wild, exhibit these cyclical changes as well as breed only during favorable seasons. These mammals are therefore called seasonal breeders. Sheep, for instance, have spring as their favorable season for breeding. Breeding during spring allows for the birth of lambs at an optimal time. That is, when the weather is warm and there is plenty of grass and water available in nature. In human beings, on the other hand, the females experience the menstrual cycle and are active throughout their reproductive phase. They are therefore called continuous breeders. The end of the reproductive phase in living organisms coincides with the beginning of the senescent phase, during which we see changes such as senility, non-existence of the menstrual or estrous cycle, and slowing of metabolism. Senescence ultimately leads to death. Interestingly, the transition between the three stages is controlled by hormones as well as environmental factors. For instance, the release of the hormone estrogen triggers the menstrual cycle in girls. Moreover, girls living in tropical regions experience menstruation at an earlier age than those living in temperate regions. In most organisms, the reproductive phase plays a crucial role as it ensures continuity of life. The birth of an offspring is perhaps one of the most important and joyous events in the life of a sexually reproducing organism. This event, however, is a culmination of events such as the development of the zygote and embryogenesis. Both these events, incidentally, are the mainstay of post-fertilization, the third stage in sexual reproduction. Did you know that every sexually reproducing organism begins his or her life as a zygote? Depending on whether fertilization is internal or external, the zygote is either formed inside the organism or outside in an external medium such as water. After its formation, the development of the zygote is largely influenced by its environment. After its formation, the development of the zygote is largely influenced by its environment. Take the case of organisms such as fungi and algae, whose zygotes are formed either in water, rocks, or soil. These zygotes develop thick walls that are resistant to desiccation as well as environmental damage. However, an unfavorable environment forces the zygote to undergo a period of rest and start storing nutrients before germinating. The environment also influences the germination of the zygote. Apart from the environment, the development of the zygote is also influenced by the life cycle of an organism. If the organism has a haplontic life cycle, as in the case with Chlamydomonas, you will notice that the zygote divides meiotically and forms haploid spores. However, in the case of a fern, a pteridophyte with a haplodiplontic life cycle, the zygote divides mitotically to form a sporophyte. After the initial development, the zygote slowly starts metamorphosing into an embryo through a process known as embryogenesis. During this process, the zygote starts to divide mitotically, which results in an increase in the number of cells. Simultaneously, the zygote also undergoes cell differentiation, a process during which groups of cells go through modification to form different tissues and organs. For example, the cells of an embryo differentiate to form nervous tissue, which later develops into organs such as the brain or spinal cord. Embryogenesis, which occurs in almost all animals, can take place inside or outside the body of the female. In oviparous or egg-laying animals, such as reptiles and birds, the process of embryogenesis takes place outside the body of the female, that is, in eggs laid by these animals. The eggs, covered by a hard calcareous shell, are usually laid in a sheltered place such as pens and treetops to keep them safe from the prying eyes of predators. Once the incubation period is over, the young ones hatch from these eggs. In the case of viviparous animals such as mammals, the process of embryogenesis occurs inside the body of the female. The embryo derives all its nourishment from its mother and begins to develop into a young one. After attaining a certain stage of growth, the young one is ready to emerge from the body of the female. Did you know that the young ones of viviparous animals have a better chance of survival than the young ones of oviparous animals, as the body of a female offers better nutrition and protection than an eggshell? Apart from animals, the development of the zygote and embryogenesis occur in flowering plants as well. Interestingly, the formation and subsequent development of the zygote inside the ovule triggers the withering of other parts of the flower, including the sepals, petals, and stamens. The pistil, which contains the ovule, however, remains attached to the plant. During embryogenesis, the zygote develops into the embryo, whereas ovules develop into seeds. The ovary, on the other hand, develops into a fruit. The fruit is covered with pericarp, a thick wall that protects the seeds and the embryo. After the fruit is consumed, the seeds get dispersed by wind, water, or animals. These seeds later germinate into a new plant. Thus, in both plants and animals, the formation of a new life hinges on the development of a zygote and embryogenesis, the two post-fertilization events. Sexual reproduction, which is common in higher plants and animals, is a far more complicated and and elaborate process